Today, I will be talking about the lytic cycle. The lytic cycle is relative to the lysogenic cycle, which in conjunction make up a grand part of the reproduction of viruses. I would describe the lytic cycle as having 4 main steps. 1. Adherence. 2. Insertion. 3. Replication, and 4. Exit. Adherence is where the bacteriophage adheres to the outside or surface of a cell. Insertion is where the bacteriophage integrates its DNA components, which it holds in its capsid, and places it inside of the cell. Replication is where the bacteriophage's DNA commences the taking over component of the lytic cycle, where DNA replication takes place and produces new bacteriophages inside of the cell. Lastly, exit is where the bacteriophages inside of the cell are to exit the cell and proceed to hunt on other cells and replicate through the same concept. This cycle relates to the mechanism by which bacterioviruses, also known as bacteriophages, reproduce. The lytic cycle is like a leech on the human body. However, instead of depriving the inside components, a bacteriophage will integrate itself in a cell and take over what is inside of that cell. To begin the lytic cycle, wherever the occurrence of taking over the inside components of a cell takes place, the predator per se, the bacteriophage, has to locate the site of its prey, the cell. Once the predator has located its prey, thus once the bacteriophage has located a cell to take over, the bacteriophage places its tail fibers on the cell. After this, the bacteriophage integrates its own material that it was holding in its capsid into the cell. The recent material that went into the cell will commence the taking over component of the lytic cycle. In this part of the lytic cycle, genome replication and translation will take place, as the material that the bacteriophage integrated initially was the bacteriophage's DNA. The reproduction process is ready to take place. Now that the DNA of the bacteriophage is inside of the cell, it will replicate itself among the cell's own DNA material. Thereafter, for their development, the creation of bacteriophages will take place inside of the cell. This part of the lytic cycle would be scientifically recognized as autoassembly. A number of bacteriophages are now existent in the cell, and the next step is for them to exit the cell where they can all go hunt on other cells, and therefore reproduce once again.